So that's helpful. To look at what? I don't. Oh, wait, right there. Does that work for you? Okay, cool. You just want to, you can sit in the back if you want. There's chairs back there. Okay, so yesterday we left off, we talked about how, through this meiosis, we do create a huge amount of variation in our gametes, in our sperm cells, in our egg cells. And that variation is important because that's what natural selection, that's what's going to determine what the species looks like down the road in generations to come. Does it adapt to the environment? If you have global climate change, then suddenly the environment has got huge changes in weather patterns. Sometimes it's really hot in January, for example, where it's 37 right now. That's not too bad, but it's supposed to be up to, 50 or something this weekend. And you're living and you have a big fur coat, that's probably not as good as having a thinner coat. So those variations will be helpful because if the environment changes, then you end up with some percentage of the population that will probably be well adapted to that environment. In terms of meiosis and in terms of chromosome number, so we know there's a diploid number, which is 2 sets of homologous chromosomes, and there's a haploid number that is 1 set of homologous chromosomes. There are times where there are really large-scale changes in the number of chromosomes that you see. And oftentimes when that happens in human beings, it is not good at all. Humans and other mammals really don't do well with, for example, an extra chromosome being added in. Or a chromosome being taken out. So in other words, taking our diploid number. What's our diploid number? 46. So changing that number from 46 to say 45 or 47 usually doesn't happen. And meaning that it is a spontaneous miscarriage before there's ever any birth or anything like that. It just doesn't, quote, take. Okay, so sometimes when a couple might have problems getting pregnant, that might be one of the reasons, is because something is happening and that chromosome number isn't what it's supposed to be. Plants do a better job than mammals do in tolerating that chromosomal abnormality. But let me actually go back to 47 for a second, because there is one 47 that we do see in humans. Anybody know what it is? I heard it. Yeah, Down syndrome. I'll just write Downs. And Down syndrome is where you have, it's called trisomy 21, because there are 3 21st chromosomes rather than 2. And we don't know a lot about why this results in the problems that it does, but it seems to have to do, again, with the idea that our cells don't work well with an abnormal set of chromosomes. They don't know what to do with that. And so with somebody with Down syndrome, again, environmental can kind of play a role in this as well. You see some people that have Down syndrome that are pretty high functioning and able to live independently. And then you see other individuals who are severely, that chromosome difference severely affects them. They may not be able to even feed themselves in some cases. And then there's a range of abilities in between. So having Down syndrome isn't just, it isn't you're going to have X, Y, and Z. You might have X, but you might have a little bit of Y, and then you maybe don't have Z at all. The symptoms and the variations are pretty big. like I said, plants do a better job of it than animals do, than mammals do. But let's talk about first about how this happens. Um, During meiosis, we have homologous chromosomes that are going to line up next to each other. And I'll just go down to the. I don't need to draw that because there's a better picture here. So here is our meiosis diagram, and starting up here at the top, I have meiosis 1. And remember, meiosis 1 is reduction division. So that's where the chromosomes are split and the homologous chromosomes separate from each other. So the homologous chromosomes line up next to each other. So remember, homologous chromosomes code for the same traits, but might not give you identical information. So you could have a widow's peak on one, and you could have no widow's peak on the other chromosome. And when they separate out, now one cell has a widow's peak, and the other cell doesn't have the widow's peak. Okay, that's reduction division. So when that happens, if those chromosomes are not split apart, spindle fibers will attach, hopefully, and they're going to pull those homologous chromosomes apart. Sometimes they don't. Sometimes, for whatever reason, we get this thing called non-disjunction. And in this cell, we're seeing that there's two chromosomes that are actually going to go over to this new this new cell. So instead of having two sets and two sets, let's do it like this. I have two sets here and I have two sets here. Or I should say I have sisters. I have three here, and I only have one in this one. And that's shown in this next diagram. You can see the 1 and the 2. Okay. Then the sisters are going to split during meiosis 2. Well, now the sisters split, but you're splitting them up so that these two cells at the end of all of this are going to have +1. They're going to have one extra chromosome because those homologous chromosomes were dragged apart at the very beginning. And same goes for these guys. These are messed up as well. But they're going to have one less chromosome. Okay. So this could be an example of R47 over here in these two. And then these two could be our 45. So if it happens during meiosis 1 is actually the worst because every one of your cells is going to have the wrong number of chromosomes. It can also happen in meiosis 2. So in this this rightmost picture, non-disjunction happens again. But now, because it's only it's happening to the sisters, and the sisters don't get pulled apart evenly, rather than the homologous chromosomes. So it's only in the second division. So that means that one cell has a +1, and this other cell is going to have a -1. But that leaves you two cells that are going to be okay. They'll have the right number of chromosomes. Yeah. Here.
Can this happen in mitosis as well?
It can happen in mitosis as well. Yeah. Well, the problem with mitosis is it's not usually as big of a problem because that cell cycle and those checks, those protein checks, the CDKs, because once a gamete's a gamete, there's no checking going on. Not until it's fertilized or does the fertilizing.
So it only affects like the gametes, like the other.
It's only meiosis is only going to affect the gametes.
So the offspring would have.
But then once you get the fertilization, then the body's going to have to go through the cell cycle again and go through synthesis, and then it would be like, wait, how do I divide this? And that checkpoint at that point might say, this isn't right. We're going to do apoptosis and we're going to get rid of that cell. So that's that spontaneous abortion. You don't have it.
Oh, wow.
The cell's like, nope, didn't pass the chromosome test. Okay. Now, back to Down syndrome again. I'll talk about that for a second here. I think that's actually in here a little more. So with Down syndrome, it is increasingly likely to see a child with Down syndrome with an increasing age of the mom. Let's say that really well. Older women conceiving are more likely to have a Down syndrome child than younger women. And when are women's eggs made? Yeah. In the womb. They're actually made before they're born. Okay. And they go through some development, but they're not fully, I want to say adult, but they're not egg cells until they start being released at puberty. However, that DNA isn't fresh, right? Each one of those eggs has DNA that was produced when the woman was in utero. So they're older. The DNA is older, and they're more likely to break. And they're more likely to break inappropriately. So you can end up with having that non-disjunction happen more commonly with older women.
Can you repeat that?
Sure. So older women are more likely to have kids with Down syndrome. And the reason for that is that a woman's eggs are all produced when she's in the womb. So that DNA has just been sitting there in that egg waiting to be ovulated. And some of them might be ovulated when they first start menstruating and they're 16 years old, and some of those eggs might not be ovulated until they're 43. And if it's ovulated when they're 43, the final division of meiosis, the meiosis 2, actually happens after ovulation has occurred. So that gives a great opportunity for those homologous chromosomes and sisters to not be separated out correctly. So the non-disjunction to happen because the DNA is more fragile at that point. Sorry, that wasn't a simpler explanation. I feel like that was more less simple. Did you get that? All right, cool. And that's why genetic testing can be good to see if, if those eggs are a problem, or what's going to happen. Aneuploidy is when you have a non-disjunction. It's a cell that results from non-disjunction. And you can have a monosomy. Aneuploidy is just a general term for not the right number of chromosomes because of non-disjunction. Monosomy is when you have one less copy. So you have, there'll be one trait that you only have one homologous chromosome for. You don't have both of them. The other thing you can have is a trisomy. Trisomy means that instead of having 2 copies of a homologous chromosome, you have 3. And that would be like trisomy 21. Trisomy 21 is Down syndrome, 47 rather than 46 chromosomes. Polyploidy. Polyploidy is a weird one. So polyploidy doesn't happen often in plants, in animals at all, like almost never. But this is where an organism actually gains an entire new set of chromosomes. So that's where meiosis went completely wiggity whack, and there was no disjunction of any of those sets, and you end up with a cell that's diploid rather than haploid, and then it's fertilized by a cell that is haploid, and now you have 3 sets of chromosomes rather than 2 sets, or in some cases it happens more than once, and you end up with 4 sets. So you can end up with a triploidy or a tetraploidy. Tri just means 3 sets, and tetra means 4. It's fairly common in plants, but it's not common in animals at all.
What happens to plants?
With plants, it doesn't seem to be an issue. And I don't know why that is. In fact, there's a lot of crops that are actually tetra and tri and tetraploidy, like wheat and tobacco, I think, if I remember right. But the kind of cool thing is that they have found recently, and I don't know how many. Let's just look at the Science Daily article here. Over in Europe, they have recently, and by recently, let's see, I mean, it's still pretty recent, about 6 years ago. They have found a crayfish that lives in freshwater that is very invasive, meaning that it tends to, all the other species of crayfish, it tends to get rid of. They can't compete with this crayfish. And one reason that it seems to be that it's so competitive and so good at all this is it is asexually reproducing. You don't typically find an animal in you don't find an animal that is asexual. It's usually sexual reproduction, and so it's meiosis, sperm and egg, and the whole thing that we've been talking about. But this guy, and by guy I mean female, because they're all females. They're basically clones of each other. And that clone is a good one. It's very well adapted to the environment. And they think that it was actually a mating somehow between two crayfish imported from Florida for a trade show in Germany. And the German and the Florida got together, and somehow this asexually reproducing species happened, and it's a polyploidy.
So this is like a new species.
New species, and it's, like I said, it's pretty invasive. So what they think probably happened was somebody dumped, they brought the thing over, and the aquarium broke and the crayfish escaped, or a lot of people, when they don't want something anymore, like a fish or a turtle, they release it into the wild, which is a bad idea. But they'll release it into the Evanston Canal, right, or even in the Lake Michigan, So that happens. So it might have been something like that. They don't really know for sure. But anyway, it's.
So wait, how does it do that? Does it just like have both gametes?
Yeah, it has both gametes. There he is. She. Sorry. Well, I didn't ask her pronouns, so maybe I'm not. I don't know.
It's kind of cute.
It's kind of cute. Yeah, I think so. But very invasive. The natural, and I shouldn't say natural because it was actually natural too, but the native species don't like it. Okay, so other things that can happen. Genetic alterations that can happen. Again, talking about chromosomes. Chromosomes can, not only can you have an entire set or have one chromosome, but you can have a chunk. So you can have what's called a deletion. So a deletion is when a fragment, a piece of the chromosome is just deleted. And that could be because of damage. It could be because of odd things happening in the environment. But it's not a gene that's deleted. It's a whole chunk. So many genes are deleted. You can have a duplication. Duplications happen a lot. And they tend to not be very detrimental. You can actually, and we'll see this when we get to evolution, you can actually follow a gene and figure out kind of the age of the gene. In other words, when did it arise? When did we first see the gene in the genetics of a particular species? By how many duplications of it are there? Is there just one copy of a gene, or is there 10? And the older it is, the more times it's probably been duplicated. So that tells you that the 10 gene one is older than the 1 gene one is. Inversion is where all the genes are there, but they're flipped. So they're inverted. So they're, instead of 1 to 10, it's 10 to 1. Usually doesn't change anything either. Translocation is an interesting one. Translocation is where a chunk stays in the genome, but it moves to a different chromosome from the one that it was on originally. So it might move from chromosome 2 to chromosome 10. And that one is really interesting because I think they're going to talk about that in a second here. Maybe? No. Okay, well, let's talk about it. Oh, is it chimpanzees? Let me just Google that quick to make sure I'm right. Okay. Yeah, so a lot of people who are advocates that we and other primates are not related to each other. We're not saying that we're monkeys or we came from monkeys, but we're saying we have a common ancestor. That's what science is saying. And humans have a total of 46 chromosomes, while primates like gorillas, chimpanzees, and orangutans, they have 48. So people would say, well, that's because we're not related to them. But the really freaky deaky kind of thing is they actually have found, and I'm not sure if I can, let's see, trying to find the best picture for this. They have found all the genes that are present in chimpanzees, gorillas, and orangutans. They have found those on a different chromosome. So what seemed to happen was that extra set of chromosomes moved, translocated onto another chromosome. And that particular chromosome is much longer than many of the other chromosomes because all of this extra genetic material moved. It used to be its own chromosome, but it moved, it migrated to a different chromosome and latched onto it. Right? That's translocation. So all those genes that the orangutans have, for example, we find them on us as well. Now the sequence is different. It's not the same genetic sequence or A, C, T, and Gs, but it's still there. And it still codes for, a protein that is involved in, I don't know, something.
We have all the same genes.
We have, it's more than 99%.
More than 99%?
With chimps.
It's just in a different spot.
Mm-hmm.
So we have, we are.
But that's pretty strong evidence, yeah.
I was going to say, it's literally.
Right. But it also tells you how important some of those other genes are, right?
Right. So do we just mutate?
Yeah, in a sense, yeah. There were changes to our DNA or to their DNA that were different. All right, let me see if I want to. All right, I'm going to go ahead and stop there. You're welcome.
We're done.
Yeah.